Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing another really awesome guest involved in science, technology, med tech that's creating a better tomorrow for many people out there. On the show today, we have the honor of being joined by Dr. Brigitte Robertson, Chief Medical Officer of Adial Pharmaceuticals. They are a clinical stage biopharma company focused on developing therapies for the treatment of addiction and related diseases, specifically with a focus on alcohol use disorder, which in the United States alone, somewhere around 35 million people suffer, resulting in significant health, social, and financial costs. And we are substantially lacking in current treatment options. Dr. Robertson has nearly 20 years of academic, clinical, and pharmaceutical industry experience. She has managed global development portfolios across multiple CNS indications in biotech, in both small and large pharma, and her experience spans from development, from bench to bedside, and she's actively engaged throughout her career in advancing biomarkers and different technologies to improve translation and trial outcomes. Dr. Robertson's industry career started at GSK as a physician scientist in their Translational Medicine Center for Excellence in Drug Discovery. She subsequently went on to hold a series of senior roles in clinical development at groups like Sepracor, Neurovance, Euthymics, and at Shire, Takeda, where she served as a VP and the Therapeutic head of Neuroscience. She also serves as the Chief Medical Officer at Humanity Therapeutic of the development of their small molecule portfolio for treating neurodegenerative diseases. Dr. Robertson completed residency training at the University of Colorado Health Sciences Center, followed by a fellowship in child and adolescent psychiatry. And she also did a second postdoc research fellowship sponsored by the NIH in neuroimaging and psychopharmacology, University of California, San Diego. In addition to that, she's held faculty positions at CCP Children's Hospital in San Diego, USCD, and Duke. A lot of really cool topics to get into today, and really honored to have her with us. Dr. Brigitte Robertson, thank you so much for taking time to come on the show.
Thank you for that really great and comprehensive introduction. I couldn't be more excited to be here today and to talking about what we're doing at Adial. It's a really cutting edge program, and it kind of brings together more than 20 years of my experience across both academic and industry research, and really what I would say, early seeds that were planted and developed my passion for the use of technology to objectify what we're doing in CNS and objectify our endpoints, and if you will, legitimize or provide that before and after photo, like an X-ray is for the size of a tumor or a lung cancer for CNS disorders. I always knew from the really the very early experiences I had in residency and internship that mental illness, as they called it back then, but even neuropsychiatric illnesses resided in the brain, which is an organ like any other organ, and that it deserved the same scientific rigor and attention and empathy that other illnesses did as well. So that's kind of where my passion started. I've always been very, very curious. My research started actually in undergrad and then was just really lit on fire through residency, working with Marty Wright in a neuroimaging lab and understanding that these illnesses and disorders, especially in CNS and psychiatry are neurodevelopmental,
Hmm.
meaning they have biopsychosocial elements that, in a dynamic way, interplay to manifest at different times in different ways in different people, which means they respond differently to interventions. And that was the other exciting thing with the neuroimaging and technological advances in technology to look at, you know, the brain while it's functioning, uh, neurocircuitry, at the same time being able to look at the biomolecular components and the genetic components to then design and have more precision around the treatments. And I think that means as well for psychotherapy, because we also know that one size doesn't fit all for psychotherapy as well, especially in alcohol use disorder, which we're kind of focusing on today.
Hmm.
But just really, I think AUD is one of the most complex disorders and most misunderstood disorders. So it offers a opportunity for us to really bring these different concepts together and how the multiplex interplays to where we are today at Adial with a
Hmm.
really exciting precision medicine program for AUD, probably the first and only one of its kind, and really started over 10 years ago. So,
Hmm.
yeah, you could tell. I'm super pumped about this program and the opportunity to be part of something so ahead of its time and just so important.
Yes, say a couple more words about that because again, I when I was going through, again, it's a really fascinating portfolio that you have. It's even pre a dial, but looking at some of the stuff that you were involved in, everything from ADHD, pediatric anxiety. I did not know that children could have strokes, and you were involved in childhood stroke. I had to look up what comorbid oppositional defiant disorder was all about, but a lot of different things. I read an interview that you gave a little while ago about, despite all of that, and we have a lot of interesting therapies in development, addiction, not as much. And I'd love to sort of just get your perspective on that. I mean, sort of being out there in the mix of all of these neuropsychiatric disorders, some kind of large, some sort of specialty, but nonetheless, and then sort of addiction over here, not a lot, as you were mentioning, happening in terms of novel therapeutics. What was sort of the environment like that you saw as you made your way through all these interesting areas of the psychiatric world? As what's going on here? Why
It
isn't
is
anything
like
happening? Yeah.
that uh, you've just honed in on like something so important and um, kind of one of like those Oprah aha epiphany moments for me as I was preparing and thinking about what to talk about in our podcast today. And that's when I realized I had not done a lot of specific work in addiction, and it kind of caused me pause. And I was like, well, I'm very close to it in so many ways, working with the stimulants and controlled substances.
Yep.
Uh, so I had to work a lot around understanding the potential abuse liability of these drugs and get them scheduled and
Yep.
like even globally in Japan where stimulants are actually still illegal. So I was able to get Vyvanse approved there because it's a prodrug and it doesn't turn into a stimulant until it gets into the body. But lots of wheeze and turns. And as I started thinking about this, I realized, well, when did addiction come to the forefront as a specialty even? And it wasn't included in the DSM or identified as a psychiatric or a neuropsychiatric disorder slash medical illness until 1984, which is not that long ago when you really think about it. And then what's also interesting is it's not housed in the regulatory agencies with other neuropsychiatric disease.
Yeah.
It's in the section of anesthesia, which sort of popped up around um, pain medications. And those were the first areas where addiction started revealing itself in opiates and the managing medical pain and oncology, and then just what kind of happened in the public sector. In a strange way, it never found its way back into neuropsychiatry, if you will, back. And I would say right now, that has influenced a few generations of physicians, regulatory people, and even the public, because it is kind of put to the side and hasn't gotten it's gotten a lot of attention, but not the same kind of attention. So a different level of scientific rigor, because in the Department of Anesthesia or the section, it's definitely not treated the way we treat all psychiatric disorders in the field of neuropsychiatry.
Hmm.
So I would say it was kind of shocking to me, but it sort of made me then dive in a bit more to understand, well, what's happened, biopsycho socially in the field. And over the last couple of years at Adial, I've also been able to witness firsthand how that divide, if you will, has impacted drug development.
Hmm.
And there hasn't been any new guidance since 2015. They're still using a lot of old terminology, like they used to talk about alcohol abuse and dependence. One of the most fascinating things to me are the endpoints for approval for medications. Um, for example, in depression or PTSD or anxiety, the symptoms are actually part of the endpoints that support an approval.
Right.
But in addiction, for alcohol use disorder, it's completely separated. Not one of the symptoms is part of the endpoint for approval. And they also don't look at the continuum the same way. For example, if you think about diabetes or anxiety, depression, we start very early as physicians identifying the prodromes, treating early mild, moderate, and severe. But somehow the dogma or the legacy view of AA, I would say, and it being a quote, maybe a moral or behavioral driven illness or disorder, I think kind of became sort of part of the fabric or the DNA of the agency and the culture of addiction.
Hmm.
And it just hasn't gotten the same attention. And it's not integrated on endpoints, drinking criteria, managing the severity. So it's a surrogate biomarker of how much you drink, which, by the way, some people can drink more, some people can drink less. The amount isn't always directly related to whether you have alcohol use disorder.
Right.
And it used to be called abuse versus dependence.
Hmm.
So all of these things haven't been updated, integrated in the documentation, like the guidance, or in the mindset within a lot of the folks that are interacting at the agencies and in the field. And I think over the last, I would say, few years, and especially just recently when they changed some of the legislation, um, really people haven't considered that a big enough problem to take any action.
Hmm.
Last year, they did update an endpoint, which, you know, it was very exciting at first because it sounded like they were going to start focusing on harm reduction, which is more like, hey, you know, AA, some of these other, I would say most of the therapy, the dogma says it's all or nothing. You need abstinence or nothing. And I would say, in what world is that even reality, right?
Right.
I mean, in diabetes, could you say you could never eat something slightly sweet or high starch or anything ever again? Or, there's just all or nothing is not a, a realistic approach to these things. It can be like if you had an anaphylactic reaction, and I'm not saying that people who want to completely stop drinking shouldn't. They should, but there are people who want to decrease or manage uh, the potential negative health consequences or what I would say biopsychosocial
Hmm.
consequences, and there are no medications for them. But our drug is just so well positioned for the spectrum of alcohol use because it's got a tremendous safety record. It's the profile for tolerability is very similar to placebo. So when you think of risk benefit,
Right.
The other drugs like antabuse, they require abstinence before you can even take the drug.
Right.
And so that already eliminates a lot of folks who can't get to that step. And the side effects and the tolerability are really horrible, when you talk to folks. So Adial's drug, which has more of a precision approach and is targeting a genetic subgroup of people with alcohol use disorder, is super as far as its risk benefit, safety and tolerability similar to placebo. How could you argue that wouldn't be worth trying or pursuing as a treatment in mild, moderate, and or severe just severity of alcohol use?
Hmm. So, I mean,
Did I
continuing
answer your
from,
question though, by the way?
yeah,
I think that's what
absolutely.
it's just there's so much here, right?
Yeah,
There's so
yeah,
much
no,
grist for
no,
the mill.
no, it's great, and we'll unpack all of this as we continue because it's again it's a fascinating story, and again, you've taken us to this domain of alcohol use disorder, as you mentioned. It encompasses abuse and dependence and addiction and sort of a lot of the terms that we thought about in the past: mild, moderate, severe. There's this 33% or so that have a specific genetic component, and we'll touch on that in a minute. Um, yeah, I mean, so the other really interesting thing that we talked about in the past is that there's a very elegant repurposing story here behind a dial and what AD04 is, which is a low dose of Danstron. And it's funny because you used to be a GSK, I used to be a SmithKline. We both had 5-HT3 receptor antagonists for specifically sold at the time for chemotherapy induced vomiting and nausea and all that. Same ingredient, lower dose, but has nothing to do with sort of the antiemetic mechanisms. Take us a little into the world of what's going on. I mean, I know neuropharmacology is extremely complex, and
Yeah,
here this thing,
yeah.
there's the reward pathway and what's happening with dopamine and at the serotonin receptor. Take a little time with, and I know it's not absolute, but what you think is happening here with the molecule and the unique sort of combinatorial nature of what it's doing specifically with AUD.
Yeah, you're not kidding when you talk about kind of the complexity of the neurotransmitter system and how we've learned so much even about neuroplasticity over the last several years and really around receptology. And I have to say that technology allowed some significant advances on the genetic side, but at the same time on the biomolecular side.
Mm-hmm.
So I think if you remember back in the day when these drugs were first developed at the agency and based on preclinical science, the approach was, hey, we've got to saturate 80 plus receptors for a response.
Yep.
And also, drug development focused on the maximum tolerated dose versus what I would call the minimally active dose, which is kind of a newer concept. Um, at the same time, I think you could look at some of the examples of what happens with that and like take Risperdal, for example, and the dopamine system.
Mm-hmm.
We had for the first time had ligands that could bind all of these different receptors, and we're still developing ligands so that you could actually see what's going on with the receptors, how you bind the receptors and let's say what percentage need to be bound to get a certain treatment response. And when you zoom back, Risperdal had a labeling that went up to like 8 milligrams. As science advanced and we retrospectively looked at those medications, it took less than 2 milligrams to saturate all of the receptors that they were targeting.
Mm-hmm.
So the rest was really just contributing to side effects. So as the receptor field progressed, what has more recently come about is the knowledge that almost like thyroid or most other biological systems, tennis, it's a sweet spot. So, you think of thyroid in general, if it's too low, you present one way. There's a range of normal.
Mm-hmm.
And if it gets too high, you present another way, too low or too high, result in clinical syndromes that have pretty negative consequences. Well, as it turns out, there's a lot of redundancy in our human biology, and our most of these receptor systems also have a high and a low.
Mm-hmm.
So the questions you're asking are so timely because as I was joining Adial, part of my remit was, hey, let's reassess, which is this kind of what I would consider my wheelhouse generally is take a step back, look at a drug, dive in to understand its pharmacology, the mechanism, update the thinking around and integrating the neuroscience, fill in gaps as needed, reanalyze the historical data, and
Mm-hmm.
then see what holds true. Because I think your point about the dose, we're using a near microdose.
Right.
So that's something that everybody kind of scratches their head and says, really? Come on. Now, it was a concept kind of coming about in like the psychedelics and other areas. But again, it takes, 4 to 8, even sometimes higher for the other indications. Well, that's when you think peripherally. When you think centrally, um, another, learning was that the serotonin transporter was kind of the sun in neuropsychiatry and neurotransmitters and brain function. And as it turns out, on Dancitron, the genetics of the serotonin transmitter or transporter are kind of more providing a background tone or variability, but not driving the response.
Okay.
And when you oversaturate and give higher doses, CNS wise, you really don't get the same responses with a lower dose that saturates just more in that sweet spot. And we also focused on the single nucleotide polymorphisms that
Yep.
are receptor level versus transporter. So the transporter, it's shifting, it kind of says, okay, it's transporting serotonin. It's not mitigating how the receptors are responding to that serotonin. So are they really super sensitive? Do they just need a little bit and boom? Do they need a lot? Um, and what we learned are these 5-HT23 receptors, A and B. Okay, there's a few of them. They're
Right.
GCPR receptors. So they're really different than the transporter. And what happened is we realized, all right, all this focus on the transporter in this program has been not wasted, if I will, but isn't the key player. As we dived in, these GCPR polymorphisms, they're guiding how the drug binds to the receptor, activates it, deactivates it, and then the downstream effects. So when the onward trial was first conducted, they were able to find that, okay, the serotonin transporter polymorphisms that they looked at by themselves didn't drive the response up or down, kind of stayed in the middle. When you added different polymorphisms from individual, genotypes that act at the receptor that the drug acts on, 5-HT23A, B, there's some others, but A and B are the I would say the most prominent drivers. B can't do anything by itself. It's got to have A. So there's even a synergy there.
Right.
But A drives most of those interactions. And if you get too much, it kind of just fizzles out. If you get the right amount, which kind of figures into the microdosing, and also that these are very small areas in the brain in the reward system, the nucleus accumbens, the hypothalamus, which, by the way, these areas also overlapped with the areas where the GLP-1s work, and decrease
Yeah.
craving.
Okay.
One of the big things about ondansetron is it decreases craving.
Mm-hmm.
But altogether, what it does is it mitigates kind of the interaction of serotonin and dopamine and the reward response system so that when you take it to a very basic level of behavior, um, people are able to gauge better their physiologic response, they're not craving it as much. Their anxiety might be a little less in general. There's also a vagal component because it still acts peripherally, but it's very tiny. However, the brain, even though it's a small amount, feeds down to the peripheral component through the vagus nerve. So there is also a calming and less of a drive. I mean, that seems kind of basic and maybe a little primitive, but I have to say that a lot of those findings just came about in the last few years. So the fact that Onward had completed, we had all of this genetic data, mechanistic data, and the preclinical data with the dosing was, kind of interesting, but we couldn't exactly figure that out. This information kind of converged at the right time. And, we recently just completed a successful end of phase 2 meeting that, really poised us well for phase 3 registration. And the scientific plausibility wasn't questioned at all. I think it's a really rational program. So the mechanism relates to the genotypes that we're targeting,
Right.
the drug, the dose to the small areas of the brain with the right amount of receptor saturation and hitting actual symptoms.
Right.
How much it will decrease or increase drinking and the play within the FDA endpoints, that's yet to be seen. But the early data, you know, was very compelling in our analysis, which we did with a partner. We reanalyzed, we integrated machine learning, AI, to make some predictions, and that helped us rule out the original analysis from the trial showed that there was an interplay or some activity of different degrees on efficacy of 4 different polymorphisms. With these analyses and bringing the science and preclinical components together, that narrowed it down to what we call the driver of these interactions. And in fact, what added more credibility to the overall theoretical construct is that these activities at the receptor level occur before activities of the serotonin transporter. So we always thought the transporter drove downstream effects. As it turns out, the transporter activity might be the downstream effect of what's happening very local at the neurocircuitry and driven by neurotransmitters and the communication across the brain. So I I think it's just a great time, right? And that all those things came together in such a eloquent way to help us kind of see our way forward and understand the biology that's going on.
Mm-hmm. Mm-hmm. And just sort of, I got to translate that a little bit. So, at the end of the day, I have my 2 beers. I will not get the pleasure that maybe the third would normally give me. I don't have the anxiety of, oh, I haven't had the third, and sort of having the letdown or withdrawal from not having it. Can you say just a couple more words about, because you mentioned the all or nothing thing before, and great analogy with diabetes. Okay, yeah, I'm not gonna never have bread again or pasta, but on the other hand, where if there's studies that are going on for whatever heroin addiction or whatever, okay, that's not, we want nothing there. Hopefully, how is that sort of the all or nothing component of sort of the clinical endpoints been greeted by, obviously the network of folks that are involved in your clinical studies? Just say a couple more words about that, because I find that kind of interesting, again, and an important point.
Well, I think so you pointed out really well, the drivers for AUD and a lot of disorders are different things. It's the same neuroanatomy. They may have different triggers, though.
Mm-hmm.
They may have stronger or softer reactions to those triggers.
Mm-hmm.
And the mindset or the cognitive behavioral thinking about that and what tools you can use to reset when you've got your lizard brain, as they call it, in the amygdala, and you're not engaging the higher cortical thinking. It's very hard to think through and to utilize those tools and what you've learned about if you drink more than, have you heard of the 1, 2, 3 rule? It's like one drink, no more than 1 drink per hour, no more than 2 drinks at an event, and no more than 3 drinks ever on a day. So if you think of that 1, 2, 3 rule, if you're at your lizard brain and you're just anxious and you're not feeling and everything, that's when the pounding happens, and people just want to do that escape and get into that place where they feel better, and the alcohol is giving them that experience that they're seeking.
Hmm.
Now, when you're able to, let's say, modify the biology, they're able to then engage more of the higher cortical functioning, calming things down. Their body is going to feel more when they're drinking closer to what I would say, I don't want to say the greater population, but the population that maybe doesn't struggle with knowing where those different limits are. And it resets things a bit. They're able to
Hmm.
understand, they're not craving it as much, and then they're able to think about it, understand it, and feel it in a more, I'd say, normalized capacity. So they tend to decrease their drinking overall. And I would think that the push in culturally, especially in so if you take the Generation Z all the way up to the boomers, um, there's a spectrum, but
Hmm.
by and large, AA and folks that gravitate to that treatment, which is fantastic for folks that works, or if
Right.
they need something or are ordered for something like antabuse and they've exceeded that level of safety for their body and for others, right? They may have a DUI or things like that.
Right.
That's one way. But most people nowadays are looking to improve their health and well-being,
Yep.
do self-care. So let's say, you tend to drink a little bit more than what they say the cutoff is, and you could take a medication like um, ADO4, and it would bring you down so that your average drinking is just a couple of drinks instead of four, five, or six. And or if you have a heavy drinking, let's say a weekend, instead of a heavy drinking day, you don't have a heavy drinking day of five, six, or seven, and it brings those and the number of those down. But
Hmm.
it's
Hmm.
not necessarily seeking full abstinence. Not
Yeah.
saying that some people don't actually become abstinent, and they did in our trials, but I would say it kind of fits both sides of the spectrum where depending on how severe and the treatment goals of the individual, they may be able to become abstinent and or decrease their drinking into a non-harm, sort of capacity or intake. Have to think about
Got it.
it. Grams of alcohol, drinks
Right.
per
Right.
day,
Right.
those
Right.
kinds of things. And it's greater than four drinks a day for men, greater than three drinks a day for a woman, on average, and then so many days a month, let's say 8, 10 days a month, that's considered heavy drinking.
Got it.
So we're seeking to bring that down into the non-harm reduction or the non-harm area where you don't have the risk associated with cardiac and liver uh, consequences.
Yep. Yep. And that's when we get into a
Is
lot of
that
problems.
I hope that it's a lot.
With
It's a lot
Yeah,
to
no,
take
yeah,
in.
no,
It's
no,
taken
no.
me a lot
Again,
to start putting these things together because even the terminology has changed so much.
right, right.
And
No,
the terminology at the
yeah,
FDA versus what we talk about,
right.
two beers, like I still hear people saying stuff like, I don't drink hard liquor, I don't do this or I do that, or I don't have cravings. So what's happened is the agency is not linked into the DSM on endpoints. At the same time, I mean, everyone knows that drinking is a component of this, but when does it turn into a disorder or an illness?
Right.
And the updates in the DSM and how that works, even with physicians and billing and all of that, they're all disconnected. So I think the public right now is kind of confused. And I think a lot of care providers are a bit confused as well. Like, what's the goal now?
Mm-hmm.
And our goal is to bring the levels of drinking below harm reduction
Got it.
or below
Yep.
harm. So we're looking to reduce the levels below the risk of cardiac and physical consequences and psychosocial consequences as well.
Mm-hmm. Mm-hmm.
Also, the other thing that I think is interesting just between you and I is uh, to decrease the suffering. Like I think in neuropsychiatry, we're even pain started that way. to decrease suffering, where the FDA endpoints, nothing about any of those kinds of things are part of the goal at all. It's just the amount that people are drinking. So they're used that as a surrogate for everything else. And I think that's a little bit of a disconnect because we all know people who don't have cardiac, liver disease, drink more than 3 drinks a day, and they function fine. They haven't had any untoward biopsychosocial consequences. We also know people who don't seem to drink that much and really get into trouble. So, there's a spectrum there, and part of that is the genetics as we're learning as well.
Yeah. Yeah. And yeah, I mean, and along those lines, so to sum up so far, novel mechanism, orally bioavailable, focus on reducing heavy drinking, no requirement for abstinence, and a unique companion genetic biomarker focus. So take us now again and take your time with this one, because we previously had the phase 2b results, and that was around 300 patients in that study, looking at things like drinks per day and heavy drinking days and so forth, but you also have another phase 3 planned. To take a little time to sort of what's happening here with the program, because you're right on the cusp of really big things, and love to hear sort of where this is all going now, from
It's you're right. We are on the cusp of it's a really I think a pivotal moment for the field, and especially for Adial right now. I mentioned that we kind of did this reassessment of the large scale trial that you're talking about, Onward, and that was the data along with some other data as well, and studies that we reanalyzed, kind of in a meta-analytic way, but specifically with the advances in technology we talked about and incorporating AI and machine learning. We worked with a partner, Cytel, to do those, I would say, the historical reanalysis of that data. And the questions you brought up, they're so spot on, because I think some of the questions from investors, analysts, from the agency had to do with what you mentioned. Okay, well, what's really happening mechanistically, or it didn't connect for folks. And the truth is, we didn't have the science had not advanced far enough to make, very firm connections mechanistically. People didn't understand the dose, and I think including the FDA. So we also incorporated dose response modeling, PK, looking more at the PK and the receptology. So that also informed us more around the dose and how that, we had good empirical evidence, but it didn't make sense to a lot of folks. And so now we brought those worlds together. At the same time, because there were 4 different polymorphisms identified that may or may not be having an effect, and the serotonin transporter is in the mix, depending on the downstream effects, you can see it's one of those if-then scenarios. So one of the suggestions we had when we interacted with the FDA was to consider an adaptive trial design.
Okay.
And along the way, with this new work and the analysis, we were able to, as I've been trying to explain, it's very hard. I probably should have brought some diagrams, which will make it much easier. But now that we understand the interplay between the genotypes, the mechanism, and response better, we're able to find that the driver was the key genotype was the 5-HT3A-G, we call it, plus. So if you carry that polymorphism, no matter what others you have, it's driving the response, but the others may enhance it a bit or decrease it a bit. So that's the driver. The others that are enhancing are part of what we're calling our biomarker positive group. And in our adaptive design that we proposed to the FDA recently, and as I said, we had I think, a really productive and constructive meeting. We had a lot of discussion about some of the things that you brought up today, but we got alignment and all the key components of the the trial. We will be submitting the protocol, the adaptive design protocol, our SAP, and our charter, for the interim analysis. So
Mm-hmm.
it'll be adapting on all comers will come into the trial. They'll be stratified around biomarker negative and biomarker positive. And at a certain point, there'll be an interim analysis to see the different responses of those genotypes in the positive or negative group. And then depending on zones of it's conditional power, meaning so if you have if the results of the interim analysis after about, let's say 50% of the subjects are enrolled in one of those arms, let's say they have a very low chance of separating for placebo, that arm may be dropped. If they have
Okay.
an in-between chance, we'll also be looking at PK, PD, and other things to confirm the directionality. And we'll also consider, we'll be doing a sample size reestimate to consider whether, well, if we add a few more subjects, would we separate from placebo at the 0.05 level required for approval? So it's a very exciting trial design. It's cutting edge, as I think it's the only one ever done uh, for a pivotal trial. And you know, we we're ready to go on it. I think the it's also, I would say, very efficient because we can make early decisions faster.
Mm-hmm.
So, if you're thinking of investing, there's earlier exit points. There's places where you could double down or make more investment earlier, things like that. Um, and the overall program, it also accelerates the overall program. Uh, we would, after the interim analysis, we would then decide based on those results to launch the second pivotal trial.
Mm-hmm.
And along the way, there's always an open label extension, as you know, for the safety data. So those all three would finish about the same time, and then we would plan the NDA after that. We don't have a perfect idea right now about the enrollment uh, speed, but we would be looking for these trials to go fairly quickly, especially the all-comers arm,
Mm-hmm.
because that's a huge population.
Right.
And so we ought to know fairly quickly whether ondansetron or AD04 works in the all-comers, and then that arm could drop off pretty quickly as a futility analysis. Then the others would kind of continue to prove the hypothesis of the selected genotype. And I think the rest is pretty straightforward. We were we discussed a lot as well because at this low dose and near microdose, there's no information out there. You still have to check the same boxes to show that it's dose proportional,
Yeah.
um, linear, and all of those things. And like I said, the possibility of using such a small dose. So we were able to align with the agency on that and also on all of the, we're not going to need to do additional safety studies like QTC and such. We've got to provide the written feedback back on that and the construct because those studies were done at higher doses and we're still low. We're not even
Right.
approaching the level of risk. So we just have to submit those and tick those boxes and then conduct the trial. As we conduct the trials, that will fulfill the overall package for approval. And I don't know what else. The rest is just there's so much at the same time. But once we get those trials started, it kind of follows the routine path, right?
Mm-hmm. Absolutely. Yeah, I mean, it's again, I can see why you're there, with your broad expertise in designing and what is really, you really have to think through how to do this. I mean, this is not, traditional your clinical trial as you're saying, but clearly, you're thinking through everything, and obviously the agency always asks for other stuff, but nonetheless, I think you're covering quite a few of the bases here, and again, it's as you're pointing out. Aside from an interesting clinical trial story, it's still a great repurposing story, a great pharmacogenetic story. So, a lot of really neat stuff. And I look forward to seeing how the things move forward there. I wanted to come back, to one of your specialties, and just to sort of connect it up here because I know it's probably, we talked about AI, we've talked about the obviously the omics and sort of the precision, diagnostic stuff. Where is all this? Where do you see a lot of that going? I mean, whether it's related to alcohol use disorder, addiction, or just CNS in general, would love to get your sort of over the horizon perspective. I mean, I know, you mentioned before you've been involved in pharmacological MRI and PET scans and all sort of the interesting ways we now can look at the brain and see what's going on. Where does this all go in terms of the wearables and some of the we hear about brain computer interfaces and all
Oh,
this stuff
yeah,
nowadays.
yeah,
Take us to
yeah.
the future a bit on whether it's addiction or just neuropsychiatric,
Oh,
neuropharmacological
God,
stuff.
it's
Go ahead.
incredible. So first of all, I just want to commend you because you really have a good grasp in kind of seeing how these things interplay and how complicated it is. Because one of the things that also has come up is that we've been really thinking about is the psychotherapy component
Yep.
required by the agency for addiction drugs, but not other drugs. So we're also kind of figuring
Right.
how that would work out. And in that perspective, I've been realizing that when you use the word blunt instrument versus precision, we've been kind of blunt across the board, even in our use of technology, the use of therapy, the different treatments. And we throw around words like blunt or precision and one size fits all. And I think psychiatry is neuropsychiatry because these are also we're finding, it's a watershed. This same areas of the brain that mitigate Parkinson's that looks like a motor disorder are the areas of the brain that mitigate the same symptoms that are in depression when you're not active as much, you're
Yep.
not thinking as clearly and all those things. And it's a different process, like I said, but it's the exact same neurocircuitry. So my sense is that the precision is just going to multiply exponentially. And with the AI and learning, integrating all of those modalities along, with the real-time data, I think we're going to be able to get precision much more closely to what oncology is having and maybe more. We're looking at things, for example, there's a couple of studies going on for real-time data looking at, like, we would use actigraphy, for example, for following motor movement. And,
Hmm.
you can pick up tremor and all of those kinds of things too for side effects for meds, cognitive
Yep.
effects using neuropsych testing. But they have a house and it's wired with infrared. So they're monitoring patients and normal subjects with different illnesses and combining all of that information. So everything they're doing the whole time, along with their heart rates
Hmm.
and their respiration rates and their biomolecular chemistry is all being kind of integrated systematically so that we're learning, so much about how the disorders and diseases manifest, what's happening at the biochemistry, which things overlap. So,
Right.
like this medication, when you understand the mechanism and what things overlap, then you can look at this broad library of medications where they've just been categorized as like, oh, antidepressants. But really, a lot of those antidepressants have like 8 mechanisms. So
Yeah.
they might help something over here in oncology or in Parkinson's or Huntington's. And so we're seeing a lot of that cross-pollination. Um, I did something recently with Fitbit. You can actually request Fitbit data,
Okay.
de-anonymized, and using Fitbit data and identifying people in the Fitbit database with
Uh-huh.
certain criteria, I was able to then do an analysis and actually find a cardiac biomarker that fit with that population. And then we could use that biomarker to see what our medication did. And was that not just correlated with diagnosis, but also with treatment?
Mm.
So that's where it's going. I think our ability to diagnose and treat these diseases and disorder are going to be way more precise and individual, and including the psychotherapy, because you know, for example, in AUD, they did huge studies, $135 million plus studies, trying to match psychotherapies and look at all different behavioral therapies, AA, cognitive behavioral, these things. Some people responded very good. Some people did not respond. Others got worse.
Mm.
And none of those treatments separated from placebo either. So is there a synergy? Is there not? How do you know what's the right therapy with the right person for the right illness? And I think that's where we're headed. We'll be integrating these, big metadata, big data, with refinement, to bring it down to an individual, even for like we know right now, like even for side effects, we've already been doing that with the SIP system, for drug-drug
Sure,
interactions
sure.
or UGT1A1, things like that. And, it's pretty great that you hopefully, when you're growing up and going to the doctor, you'll have a basic idea of, hey, these are some of my risk factors. These are meds I can take, I can't take. These are where I'll have interactions. These are things I can do now to help prevent decompensation or to go a different way. Um, and I hope within all of that, that we address what I would call the environmental component. I follow the mind diet. I'm super passionate about what we put into our bodies. And, when I was a resident, we had a McDonald's in the lobby of Children's Hospital. And I used to eat those little yellow crackers and Mountain Dew for breaks. And
Ah.
I just cringe now and think, you know, should I have known? Should I not have known? But also, even as a kid, what's a Fruit Loop? What's a Cheeto?
Or what about Hawaiian Punch? Remember that
Oh
one?
my God.
Yeah.
Yeah, Everclear Punch too. But yeah, exactly. And while there's an awareness of that, it's getting better and better. I don't really see corporate industrialized America that's managing the food and what's going on in our bodies. They're using new buzzwords, but when you actually still read what's in the ingredients, especially sugar, which
Mm.
is cancer, for example, loves sugar.
Mm-hmm.
Parkinson's, a lot of these diseases, anxiety loves sugar. Anxiety is the risk factor, or is it sugar and diabetes are risk factors for Alzheimer's? So when you start connecting the dots, a lot of it is environmental, and we really need to, I think, as a society, take this opportunity with the young people, the elderly people, and just demand uh, more precision in our food sources as well. But
Mm-hmm.
well, that's my rant on that. I'll stay away from that.
No, no, no.
There's just so much grist for the mill, and it's I could never have imagined when I joined Adial. I was a little bit, I'll be honest, I'm kind of a cautious uh, optimist when it comes to looking at data and re-looking at things. And I also hadn't had a lot of experience specifically in AUD. And it was one of the best things that I've ever done. It's broadened my horizon, and I have to say the group of people I'm working with, I haven't worked with any better people. They're really, I think Carrie was on your webcast before, right?
Yeah, yeah. Last year
Yeah,
we.
they're just awesome, real, straightforward, direct, not a lot of hubba bubba. So we've just put our nose to the grindstone, and they've just really supported what I would call kind of true north, or trying to find as much the kernel of truth and the truth and the data that we could to build on or not.
Right,
When I came on, I was very clear that, if I don't see anything, I'm not someone that can get behind something I don't believe in.
right.
So they were totally supportive of that. And, to get me to get so excited about it, you would imagine the findings were pretty impressive for this genotype.
Yep. Yeah. Yeah.
Anyway,
You're definitely excited and passionate
yeah,
about
I
it.
know.
I
Yeah,
could tell
I
that.
probably said it too many times, but
No,
you can cut some of those out, okay, Ira?
no. It's important, and again, I've said it multiple times. That I was very intrigued by the story, when Carrie was on the show, and again, I keep coming back to this repurposing thing, and I think it's going to be interesting. Again, as you're pointing out, with a lot of these neuropsychiatric drugs, we know so little about how they work, and the interesting things we're going to be finding out, and the whole psychedelic thing as well. That's going to be
Oh
pretty
my God, I
interesting,
love
especially,
that.
yeah.
We'll have to talk about
But yeah,
neuroplasticity and how they're
well,
doing that sometime. But
yeah,
yeah, if you ever just want to like have a great talk and we're not doing a podcast, I would totally welcome that. I was
that's
re
right.
looking through a lot of your podcasts and um amazing. You have a resource of information and you've been a part of those interactions. So I imagine you've become kind of a resource yourself.
Yeah, yeah, I know enough to be dangerous now, but
Yeah, no,
but
I don't think so. But
yeah. But while we have you, I just so we're going to be looking forward to what's happening next as we get into 2026. Anything else, just while we have you today, that I missed? And then any public facing stuff. Obviously, you're public facing now, but conferences, talks, other things that are happening as we get into the later part of 2025 and into 2026 that you want to mention,
I
please.
think we're just lining up talks and things like that for 2026. We'll be looking to have some abstracts and presentations at some conferences on the phase one data and the microdosing, or
Hmm.
it's near microdosing. It's just a little bit off of microdosing. Also analyzing the onward trial for the new endpoints using the decreased risk level and how that reflects it's not as sensitive as the other endpoints, and also kind of pointing out some of the disconnects between the agency, and maybe thinking about it one of the conferences where we'll kind of bring the pieces together on the new analysis, what we've learned, and a little bit of an overview, a little bit like we did today, maybe less informal, but kind of walking people through what's going on. Um, as we get those nailed down, we'll send you those. You can put them up, but near term, don't have the exact venues
That's
for
okay.
those yet.
No worries, no worries. Well, we will, of course, we'll be putting a link to the website and everything we talked about today in terms of the portfolio and the news and all that. But no, it's again, I really I'm excited for you. I think it's a great story and why I love profiling it and just really wishing you the best with it and your team. Again, for everybody that is going to be listening to this episode of the show across the various podcast networks or watching it on the YouTube channel. Again, you've been spending time with Dr. Brigitte Robertson, Chief Medical Officer, Adial Pharmaceuticals, doing really amazing work to develop therapies for alcohol use disorder and related disorders. Brigitte, I want to thank you for taking the time to walk us through this really fascinating story. Thank you for everything you're up to and helping patients. And as we like to say here on our show, thanks for helping to create a better tomorrow for literally millions of people out there via what you're doing.
Thank
Really,
you
really
too,
speaks
Ira.
on.
It was such a pleasure to speak with you. I really enjoyed it. Thanks so much.
Really great having you.